We are going to discuss the excitation-contraction coupling, the molecular mechanism by which a muscle fiber contracts. The primary functional unit in a muscle fiber is called the sarcomere. And last time I had mentioned that sarx was Greek for flesh, and meros is part. Sarcomere is the fleshy part of a muscle fiber. When you look at a muscle fiber under the microscope, here's an electron micrograph, and we see this striation pattern in more clear detail. There are these dark bands and these lighter bands. The first German microscopists that identified this used German terms to depict a lot of this. And so we'll see that as I go through identifying each of these regions. There is this dark band is what we call the A band. And A band stands for anisotropic, and I band is isotropic. The lighter color band is isotropic. The terms isotropic and anisotropic are generalized terms that refer to any object, in fact. Something that is isotropic or can be tumbling isotropically or can be scattering light isotropically. Isotropic means that there is no directional preference. There is no directional preference that whatever we're talking about, whether it's a tumbling, whether it's a scattering of light, there's no preference. It scatters light equally in all directions, or it tumbles equally likely in all dimensions within space. Okay, and anisotropic implies a directionality. Okay, so if you're going to, in this case, anisotropically scatter light, you're going to be refracting that light at a certain angle. This implies certain things about the molecular structure of the material that you're beaming the light on. So anisotropic material is going to have some sort of repetitive, symmetric handedness to it. And we'll see what that means when we look at these in more molecular detail. Okay, so when they were looking at this stuff underneath polarized light, does anyone not understand what I mean when I say polarized light? Because I'll happily explain. Everybody has got polarized light down? Oh, you do not. Polarized light. So light is a wave, right? And if you were to look at this wave down the barrel, down the barrel of that wave, it's not really even just a wave. It's almost like a bead, a string of beads. All right. So the wave is going up and down in this direction, but also this direction, and etc. Okay. So when you polarize light, you're going to put it through some sort of material that has slits through it like so that when this light passes through it, we're only allowing light that lines up with that polarization to pass through it. And this light that's anti-parallel, that's perpendicular to our polarization slits, does not pass through it. So because of this, you're only looking at light that has certain orientations. Okay, that's polarized light. So when you apply polarized light to a sarcomere and you change the phase, you're able to take this thing here and rotate it. You rotate the polarization of the light, changing the phase of polarization without changing the orientation of what you're looking at. You're going to see a sort of an iridescent phase shifting in the way that this light looks compared to this. The I bands do not change the way they look, okay? Because they are anisotropically scattering light. Yes? Maybe I'm belaboring it too
They
much.
rotate and they shift the phase?
Say that again?
They're rotating the light and they're shifting the phase of the wavelength?
They're not
shifting not the wavelength, but they're shifting the phase of the
They're
wave.
shifting the phase of the polarization. So from parallel to anti-parallel. That's all they're doing.
Okay.
Yeah. All right. So they're down the middle of this I band is what's called a Z line. Z stands for Zusammen, a German word that means. You please tell me.
Together.
Yeah, together. That's right. Thank you. Because this is where the two. When a muscle contracts, this is where two adjacent sarcomeres are coming together. They're both approaching this Z line. The Z lines stay relatively fixed, and it's the point at which two adjacent sarcomeres are coming together. The M line stands for Mittel, M-I-T-T-E-L, meaning middle, and it is the middle of a sarcomere. And so these Z lines are what join adjacent sarcomeres together, and this is the middle. The M line is the middle of a sarcomere. And then there's this H band, which is this region in the middle of an A band that is a little bit less dense. It is a little bit less dense looking under the light. We'll see what all of these mean in just a moment. This area here, after we began to understand the molecular functioning of a sarcomere, this became known as the zone of overlap. So the zone of overlap is the part of the A band that is not the H band. All right? There's the M line down the middle of the H band, and then there's the zone of overlap. All of that makes up the A band. I'll just tell you now, yes?
I guess, is there a specific reason why the middle is the H band and not the Z band? Because they're repeating every other one? Is that just by convention, or is there some other reason why it's better to have the H band be the middle than the Z band?
Hold your curiosity. I will see if I satisfy it. Okay. I understand your question. So the reason we call this, first of all, let's describe what we're looking at. We have thin filaments and we have thick filaments. And we will see what those are in more molecular detail in a moment. But for the moment, we have these thin filaments and these thick filaments. And it is the interaction of thin and thick filaments which provides the contractile force. All right, one is going to be pulling itself along the other. This M line is where all the thick filaments attach to one another. Likewise, the Z line, which people have taken to mean zigzag, but it's not what zigzag means. It's Zusammen, as Yannick pointed out, means together. But this is where all the thin filaments come into contact with one another. The Z line is attached by these thin filaments. That's the wrong word. That's the wrong word. I don't want to confuse you. But by these strands to the ends of the thick filaments with this protein called titin. And titin helps to stabilize the filaments, keeps them all organized and lined up properly. We can see here that this H band is the area that is only, thick filaments. And the zone of overlap is where there are thin filaments and thick filaments together. I want to make sure that I didn't say the definitions of anisotropic and isotropic incorrectly. Something that is isotropic is scattering light. I probably did say it backwards, and I apologize for that. I'm not sure, though. It's all on record. You can go and see. Catch me. Isotropic is something that is scattering light to within a certain directionality. Is that what I said, or did I say it backwards?
You said anisotropic is directional.
Anisotropic is undirectional. Okay. Isotropic is directional. I put those terms backwards. I apologize for that. All right. It's letting my tongue get ahead of my brain. Okay, so this is not that important of a slide actually, but it's just looking at cross sections through different points in the sarcomere. And what's interesting here is just looking at the hexagonal array that you're going to see. This is giving us the maximal overlap, the maximal contact between thin filaments and thick filaments. So any thick filament is going to be surrounded by 6 thin filaments. All right? Any thick filament that you're going to find is surrounded by 6 thin filaments in this zone of overlap here. And this is the important part. The zone of overlap, as I said, is where the actual chemical reaction that's going to lead to contractility of the muscle takes place. All right? All right, so let's go through these one at a time. Thin filaments. So the thin filaments are what makes up the I band, the isotropic band, the part of the part of the sarcomere that is going to uniquely scatter light within with a certain handedness. And you can see here that it's made of F-actin, filamentous actin. Now, actin, G-actin, is a globular protein monomer that then polymerizes as a homo polymer that associates along this core protein called nebulin. 2 strands of F-actin form a helix around the central core of nebulin. There are 2 other important proteins here, tropomyosin and troponin. Some of you have probably already heard about this who've had like high school anatomy or whatever. Tropomyosin is, again, a double-stranded protein that is helical and winds its way along the surface of the F-actin, coming in contact with what we're going to call the active site of the actin. In fact, covering that active site. That orientation of tropomyosin is controlled by another protein, another globular protein called troponin. Troponin's job is to hold the tropomyosin in place and to keep it occluding the active site of the F-actin. This troponin is this function of troponin is controlled by calcium 2. All right, so when there's calcium 2 around, when there's calcium 2 in the sarcoplasm, it will bind to troponin, and troponin will roll tropomyosin off of this active site. Do you guys follow that? Do you see how there is this sort of symmetric handedness to it, and how the I band may scatter light with a unique handedness to it? We'll see then how thick filaments do not have that. So thick filaments are made of this protein called myosin. And myosin, a monomer, looks essentially like this. So there's a long tail region and then a little myosin head group and a hinge region. It's sort of like a space-age golf putter here. It looks like a little bit like a golf putter where there's a spring loaded. You don't even need to move your arms. The head is going to move of its own accord. All right? It's this head that's going to come into contact with the active site on F-actin and lead to the contraction. We'll see how that works in a moment. So these thick filaments are these large bundles of myosin. And they show a little bit of a spiral array here. However, myosin is actually anisotropic and does not scatter light with a certain handedness to it. That's just a cartoon. And then the end of this thick filament is where titin attaches, helping to keep the fibers aligned, the myofibrils aligned, and to help recoil. All right, so there's a number of other proteins then that are associated with contraction, but those are, I shouldn't say with contraction. That's the ensemble for contraction. However, taking the contraction of myofibrils within a sarcomere and translating that to the contraction of the muscle fiber, which is going to contract a muscle, requires these accessory proteins that connect a sarcomere, the mechanism of a sarcomere, to the sarcolemma, the plasma membrane of the muscle fiber, which are going to actually physically contract the muscle fiber itself. All right, and this muscle fiber is then attached to the connective tissue, the endomysium and paramysium, etc. Some of these proteins we have going along here. The one that is notable is this protein dystrophin, and this is actually the linker protein that connects the sarcomere to various proteins that are embedded in the membrane. This is notable because it is a problem with this protein that leads to muscular dystrophy, and this is why the protein itself is called dystrophin.
So, how does the surface of the, sarcolemma change shape when the muscle cells? Does it just bow out and sort of does the dystrophin kind of like extend and keep it anchored, or does it kind of fold over onto itself, or what does it do?
This is an excellent question. I've never actually seen a good animation or description of that myself, but I'm imagining that it's going to have a slightly ruffled pattern as it contracts. You're going to have the distribution of the dystrophin and these anchoring proteins, throughout the sarcolemma to be able to allow the contraction of that cell to happen. I don't have a good answer for that one. Okay, so this is the point that I was making earlier about the sort of fractal structure of muscle tissue. You have a skeletal muscle, which then is a collection of fascicles and surrounded by this epimysium. A fascicle itself is surrounded by the paramysium and is a bundle of muscle fibers. A muscle fiber is surrounded by endomysium and is a collection of myofibrils. A myofibril is surrounded by this sleeve of sarcoplasmic reticulum and is composed of the individual sarcomeres. A sarcomere is then composed of the myofilaments, the F actin and the myosin. Yeah.
Oh, at what level are like the main capillary beds that are supplying the nutrients to the muscles? Is that going to be around the
arterioles?
The
Arterioles pass through the epimysium. The capillary beds are going through the paramysium, and with small tendrils perforating down to the muscle fiber level. All right. So what is a muscle contraction? I've already sort of said this, but it's simply the interactions of the thick and the thin filaments. All right. And we're going to have these thin filaments that are anchored, and then these thick filaments are going to walk their way along the tracks laid down by these thin filaments. This is going to take a tremendous amount of energy. Because of that, there's going to be significant glycogen reserves in the cell. That's what all of this is here. These are mitochondria in green that are distributed throughout the sarcoplasm and surrounded by all this glycogen that is going to be necessary to be mobilized and to produce the ATP, which is what we'll talk about on Friday. All right, so this interaction between thick and thin filaments is what's called the sliding filament theory. So what do I have here? Yeah. You can see here as I go back and forth between these two cartoons, this would be the relaxed muscle, all right, and then here it is contracted. The myosin head groups have walked along the train track of the thin filaments. And we'll see how this works in a moment. What's interesting about this, you'll notice, is that the width of the A-band always stays the same. All right, the width of the A-band always stays the same. And in fact, the length of the thin filaments always stays the same as well. However, when you contract, it is the width of the I-band that is getting shorter. It's the width of the I-band that's getting narrower and was actually connecting. And this is why they call the Z-line the Zusamin, because it's bringing together the two portions of the two adjacent A-bands. Okay? All right, so here's an overview of what happens. There's going to be some sort of neural stimulus that comes down through the axon to the neuromuscular junction. There's going to be a synapse, a degranulation, stimulation of a chemically gated channel that's going to trigger a bunch of voltage-gated channels. That's going to have an excitation across the entire excitable membrane. It's going to pass across the entirety of the sarcolemma and then down into the perforating transverse tubules. Those transverse tubules are going to be a part of this triad with the terminal cisterna of the sarcoplasmic reticulum. This depolarization is going to cause a change in the membrane potential here, and it's going to effect a calcium release from the stores, the sequestered stores of calcium inside the sarcoplasmic reticulum. This calcium is then going to bind to troponin, which will roll tropomyosin off of the F-actin and enable the contraction mechanism that we're going to go through in detail in a moment. This contraction then leads to the shortening of a muscle fiber, which creates tension in a muscle and the effect that you're looking for. Excitable membranes is the theme of this whole mechanism here. So there are four major phases to this. I'm going to go through each of these in detail. So this is the overview for you. If you don't get everything on this slide down, that's okay, because I'm going to go through each of them in turn quite slowly. So first of all, there's going to be this excitation, and this is the process by which a somatic efferent nerve, a nerve that is meant to excite a muscle fiber, that action potential is going to lead to an action potential in the muscle fiber. All right? And then there is the excitation contraction coupling. So this is the mechanism by which that depolarization across the surface of the sarcolemma is going to lead eventually to the contraction, but it's this coupling of the excitation events on the membrane that were elicited by the nerve. It's the coupling of that excitation to the contraction events that are going to happen on the myofilaments. And then the actual contraction. There's a set of molecular mechanisms by which The myosin head group is an ATPase. It's going to hydrolyze ATP and convert that energy into actually motion of that head group, which is going to apply contractile force to the muscle fiber. And then finally, the last bit is relaxation. This is very important. This is an important step in the cycle. If you contract a muscle, you want to be able to release the muscle as well, and so there are mechanisms for that as well. Okay, so first, excitation. This happens at the neuromuscular junction. There's some sort of action potential which is going to impinge upon the synaptic bouton and cause the release, the degranulation, the exocytosis, the fusion with the presynaptic terminal of these synaptic vesicles, which are filled with acetylcholine, a neurotransmitter that is going to stimulate the chemically gated channels on the postsynaptic terminal. All right? So, and this is what this looks like here. This is the motor end plate. This would be the histology. You see these telodendria coming down to the motor end plate here, and these synaptic bouton that are filled with vesicles that are going to release their contents into this synaptic cleft. So presynaptic terminal, postsynaptic terminal, synaptic cleft is the space in between them. All right, synaptic vesicles filled with acetylcholine. Chemically gated channels right here. Adjacent to it, but not depicted, are going to be a bunch of voltage-gated channels in the sarcolemma, which are going to get triggered. All right, let's see what happens. So, step one, this action potential arrives coming down the axon to this neuromuscular junction at the motor end plate. Step two, this depolarization in the neuron causes degranulation. Here's acetylcholine being released into the synaptic cleft. Step three, this acetylcholine binds to the chemically gated ion channels in the postsynaptic membrane, thus allowing the influx of sodium, allowing sodium to travel down its electrochemical gradient. All right? Sodium's chemical gradient wants to go in. So here is a picture of that. You'd have some sort of transmembrane acetylcholine receptor. It binds to acetylcholine. It changes the diameter of that pore and thus allows sodium to pass through. All right. Yes?
So, is that technically, it looks like it's letting in sodium and out potassium, or is that happening? No, this one looks different as to where that actual travel is happening.
Yeah. So this is a chemically gated pore. This is a voltage gated pore, and again, it's just a cartoon. It's not actually showing the activation and inactivation gates that both of these channels are going to have. Yeah, so don't get hung up on the fallibility of the cartoons I'm using. All right, so once we have established a local current that is depolarizing this this membrane, that local current is going to trip the sodium channel and allow an action potential to start across that membrane. That depolarization, that wave of depolarization will travel across the entire excitable membrane. Has anybody gone to any of the videos that I have embedded in the Moodle? There's some really nice videos that depict this. If I have time, maybe I'll go to it in class, but it was meant for you to look at outside of class. There's some really, I uploaded some excellent videos there that that illustrate all of this, that animate this. Probably could save me 50 minutes with a 2-minute video there. But so depolarization causes this action potential that's going to spread across the surface of the sarcolemma, and that's what we see happening here. And then as that happens, this other protein here called acetylcholine esterase, it's an enzyme whose job it is to hydrolyze the ester bond between an acetyl group and choline, and essentially recycling the acetylcholine and taking it out of the synaptic cleft, so that these sodium channels can, the chemically gated sodium channels can then close. All right, and returning this presynaptic terminal to its original state. All right, as that's happening, there's going to be the sodium and potassium pumps trying to reestablish the chemical gradient. Yeah.
So how do the materials get back into the synaptic bouton?
There's a recycling pathway that I'm not going to take time to go through, but yeah. The choline is brought back in. There's a recycling pathway. So the next step is the excitation contraction coupling. And this is essentially the story of how the action potential that's traveling across the sarcolemma passes down through a T-tubule into the region of one of these triads. And you'll remember a triad is a T-tubule with two adjacent sort of pouches of sarcoplasmic reticulum called terminal cisterna. There's these cisterns at the end of the sarcoplasmic reticulum directly adjacent to a transverse tubule. This depolarization in this membrane right here, adjacent to the terminal cisterna, causes these voltage-gated calcium channels to open up in the terminal cisterna of the sarcoplasmic reticulum, and then release calcium into the sarcoplasm. All right. So within the sarcoplasmic reticulum, the ion that's important is calcium. It's calcium. You'll remember from the first or second day of class, I was talking about the importance of calcium homeostasis and calcium's role in muscle contraction, and this is where it is. All right. So the sarcolemma, the dynamics of the sarcolemma are dictated by sodium and potassium. The dynamics of the membrane and the sarcoplasmic reticulum are dictated by calcium. Okay? So there's going to be calcium leak channels, there's going to be calcium pumps, there's going to be these voltage-gated calcium channels. Okay. So, and that's the short story. Well, I guess the last bit of it is this calcium then comes and binds to troponin. And when that happens, when calcium binds to troponin, troponin rolls tropomyosin off the active site in the F-actin, in the thin filaments, thus revealing them and enabling what we're going to call cross-bridge formation, enabling the interaction between myosin head group and the active site on F-actin. You follow me? Is anyone's hair blowing back here? Everyone good? Okay. So that is the excitation contraction coupling. That's how we get from synapse to contraction. Okay? Now it's time to talk about contraction. At the beginning of a contraction cycle, we start having already paid the bank. We've started already having paid for the first contraction. there is already an ATP that's been brought on board a myosin head group, been hydrolyzed to ADP and inorganic phosphate, and the energy of that hydrolysis of the phosphodiester bond has been stored by cocking the head of this myosin head group back and storing that force in the spring of the hinge, okay? So we start having paid for it. Calcium comes in. Let's do that. Calcium comes in, rolls the tropomyosin, binds troponin, rolls the tropomyosin off, reveals this active site, and now we're ready for the beginning of a contraction cycle. First thing that happens is we're going to get cross-bridge formation. Okay? So this myosin head group is now able to desolvate this active site and bind. The myosin head group is able to bind to the active site on the actin. And all of what I'm about to tell you, all of these steps are a sequence of dominoes. Each step that happens is the trigger. So again, these are cartoons. I'm not going to take you into the in-depth atomic resolution interactions that are happening here. But maybe in graduate school, if you get there, it's worth your time to look at that, because it's pretty interesting how the molecular shifts that happen with each of these events leads consequently to the next, or subsequently to the next event. So we have cross-bridge formation. That formation of a cross-bridge, that interaction between the head group and the active site is going to cause a shift in the molecular arrangement of that head group that's going to allow two things to happen. It's going to allow the energy that has been stored in the hinge region to be released, and thus causing that head group to snap forward. This is where the rubber meets the road. All right? When that happens, another structural shift happens that boots ADP and inorganic phosphate off. Those are coincident. There are coincident events. All right? So head group rotation boots ADP and inorganic phosphate off, and we have the first inklings of some contraction. But we're going to go through a cycle. We're going to go through a cycle. The cycle is going to happen as long as calcium and ATP are around. ATP hasn't entered the picture yet, though. Right? We're about to add it. So when the ADP comes off, the cross bridge detaches. There's cross bridge detachment. When that happens, this reveals the potential for ATP to bind to the myosin head group. When ATP binds, it binds in such a way that that the myosin head group effects the hydrolysis of ATP into ADP and inorganic phosphate. When the hydrolysis happens, that energy is released and stored by cocking the head back once again. And we're ready, as long as there's calcium around to keep troponin engaged and keep tropomyosin off the active group, we're ready to go around the merry-go-round again. Okay? So myosin reactivation. So what does that look like? Well, here is a muscle fiber uncontracted and contracted, relaxed and unrelaxed, and looks something like that. Did I do that right? I guess I did. So you can see how the A bands stay the same width, but it is the pink I bands that are getting shorter as the zone of overlap. Is that giving people a headache? As the zone of overlap gets shorter. Yeah.
And the H bands also decrease.
Absolutely. Absolutely. Yeah. The H bands get smaller at the same rate that the I bands get smaller.
The
Zone of
is the
overlap.
same length, so.
Yeah. So, can I have a few volunteers? Not more than 3. Please, 1, 2, just come on up. Preferably people who aren't afraid to hold hands. Come on up. Okay. So get in the line right along here. Stick your arms out like this. Get way, way down there. And put your arms are going to be out like this and interacting with the other person. And what we're going to do is we're going to keep our arms straight but sort of ratchet our way up the other person's arm. Ready? Let's do it. Okay, so we're contracting. We're contracting. All right? And now we're going to relax. And we're going to slide back out. That's all there is to it, folks. All there is to it. Sliding filament theory. Okay? Thank you. Thank you very much. It's good to be active. Get the blood moving. They say every 20 minutes you should get up. Okay. How much time do I have? Great. So, how long does this last? Well, it's going to last dependent upon, the duration is going to be dependent upon 3 things. First of all, the duration of the neural stimulus. How quickly is the acetylcholine esterase going to be able to break down the acetylcholine, recycle it, and allow those channels to close, the chemically gated sodium channels to close. It's also importantly, so this is at the excitation phase. All right. Here, this is at the contraction phase. That's going to be dictated by how much calcium is available in the sarcoplasm. Is the sarcoplasm, is the T-tubule no longer depolarized and have the calcium channels, voltage-gated calcium channels in the sarcoplasmic reticulum closed, and the pumps that are pumping calcium back into the sarcoplasm, have they caught up with the process and sequestered enough calcium? So how much calcium is free in the sarcoplasm? And, of course, do we have ATP for this to happen, right? So the bulk of Friday is going to be talking about this and how we're able to maintain a steady state of ATP for this process to go on for a length of time. The different methods by which the cell has of delivering ATP to the cells for contraction. So finally, yeah, Jacob.
So like if you've reached a point of like calcium equilibrium, which I don't know if you ever reached, but that would be bad, would you then just cramp up? Because it's like you have so much calcium released.
So I like to ask a question about this actually on the final. There is fatigue. You're getting at muscle fatigue. There is this thing called the rhyatidine receptor, which is a calcium channel that becomes leaky, preventing the sarcoplasm from sequestering all of its calcium to allow a contraction to actually end. I don't know if I'm answering your question, but
basically the contraction doesn't end if you have too much calcium out there.
That's right. Yeah, you can end up not allowing the muscle fibers to reach their the preferred length, and we'll get to that in just a couple slides here. Probably not today, but let's talk about the last phase here, relaxation. So here's the acetylcholine esterase, and this is a gross simplification of the recycling pathway that happens. There is a fairly sophisticated recycling pathway that happens for bringing the choline back into the synaptic bouton for resynthesis into acetylcholine. So that's the first thing that has to happen is the removal of acetylcholine from the synaptic cleft. When that happens, calcium gets resorbed into the sarcoplasmic reticulum by the calcium pumps that are there. Troponin loses the calcium. Tropomyosin rolls back onto the active site, and we are now no longer able to have cross-bridge formation. ATP, this is important, ATP is needed for muscle relaxation as well as contraction, because this process of sequestration of calcium in the sarcoplasmic reticulum requires ATP. All right? So you have to have a source of ATP on hand to be able to reset that muscle for a subsequent contraction. All right? Yeah, Yannick.
So you don't mean like when you're relaxing, you're not just using ATP as well. It's just for relaxation.
It's for the relaxation phase of a contraction. Yeah, to reestablish the electrochemical gradient of calcium in the sarcoplasmic reticulum. You're going to need some ATP. All right. So we're clearly getting close to the bewitching hour. This is, let me see here. Yeah. Let me just point these out here. We have various elastic forces, the connective tissue that's dictating the length of the muscle and helps bring that sarcomere back to its resting length. There's the action of opposing muscle fibers. So I'm going to use my biceps, and then I'm going to use my triceps to extend my arms to help bring the biceps back to their resting length. And then, of course, our friend gravity can help with that as well. So this is just a summary of everything that we talked about today. And, yeah, we'll start with rigor mortis next time. We'll start with rigor mortis at the next class period. Are there any questions? All right. Cool mechanism. Cool mechanism.
Are we going to talk about smooth muscle?